So welcome to the last lecture in basics of molecular modeling of drugs that is actually focused on case studies. So I would like to show some examples of real drug, how they were developed using the methods of molecular modeling, and maybe also some compounds from the development that actually do not become real drugs. But the molecular modeling methods were used in the development. In the beginning, when molecular modeling started, the scientists thought that it will be very useful and they will be able to design the drugs only using molecular modeling. However, it proved that it is possible to do it only for lead compounds. And from lead compound to candidate, there is further drug optimization necessary in the mean of synthesis of multiple compounds and screening of multiple compounds. So it means that molecular modeling is only one of the powerful tools in the hands of pharmaceutical chemists, done by computational chemists. The design process is considered in pharmacodynamic properties as well pharmacokinetic properties. And in the figure below, you can see the increase of the term virtual screening in the beginning of the first decade of the 20th century. So let's focus on typical workflow in drug design. Already in my very first lecture, I said that identification of target biomacromolecule is the first step of targeting any diseases. It is necessary to clone the target, which is usually a protein, to express it and to get it in a purified form. And in laboratory, we have to establish an assay which helps to test the compounds, if they are active or inactive. And we also need crystallographic structure or 3D structure of the protein. We can generate it most often by X-ray or by NMR spectroscopy or by cryo-electron microscopy to get the coordinates of the target, and we can use them in structure-based design in silico. So the right part of the plot concerns in silico chemistry. So at the stage where we are getting the lead compound, which means any active compound against the targeted protein, we can verify in silico, for example, from our database of compounds, what compounds have the chance to become a lead compound, which is less financially demanding than screening huge databases in laboratory. So we can use the structure-based drug design for getting the lead compound. We can use it also in optimization. So the lead compound usually doesn't have chance to become a drug, but it requires structural optimization to get very potent compounds. So we need to increase the activity, increase the selectivity, optimize pharmacokinetic properties, and so on. And the compound that is that is produced by this process is called drug candidate. And this compound has already chance to become drug that is registered in the market. So on the right side of the slide, there is described who else is involved in the drug design process. It starts with computational chemists, a synthetic chemist, a medicinal chemist. We also need the molecular biologist. We need the crystallography and NMR specialists. So it is a huge collaboration, coordination of these all disciplines. So just to revise a little bit, what specific methods can be used? So during the target identification and validation, we use so-called drug ability assessment. For example, the company Schrödinger sells the software for judging drugability of protein. So protein is judged from the point of ability to bind to any specific ligand. So not all proteins are able to become target of drugs. So there are sophisticated software that are able to judge it or score it, actually. During the hit generation, like generating active compounds against a specific target, we use different methods. For example, property and structural filtrating. So the typical filter is, for example, a Lipinski rule 5. We use also clustering methods. So, for example, the software Scaffold Hunter, you do not need to memorize the softwares. It's just for illustration. Is able to cluster together groups that have some common structural features. And group the compounds in a hit list. So this is very helpful when we deal with big databases of compounds. And we found some of them active. So we can explore the hits based on the similarity clustering. When we get the hits, we can optimize the hits to lead. So we can again use molecular modeling softwares and exchange, for example, functional groups, exchange the scaffolds, and prepare virtual combinatorial libraries. So we can modify in silico very active compounds to get a larger library, and we can screen again in silico and predict if the predicted ligands, like predicted compounds, are able to bind better to the target than the hit compound. And of course, we use structure-based drug design during lead optimization, like judging the pharmacokinetic, toxicity, bioavailability, oral bioavailability, metabolic stability, and so on. So during all these steps, all these processes, we use the in silico methods and different sophisticated algorithms. Just to illustrate the drugability of the target, so There is a online database where you can find different drug targets, and on the right side there is an output from testing druggability of bromodomains. These are proteins that can be used in cancer treatment as the target. However, not all bromodomains are suitable. So when we look on the output, the red highlighted bromodomains would be suitable as a drug target. The yellow ones are so usable, and the white ones are not suitable to become a target in a disease pathophysiology. Here is an illustration slide focused on clustering of ligands. So we talk now about hit compounds that have shown some activity or can show some activity. So we usually use the filtrates based on molecular property descriptors. So it is molecular weight, lipophilicity, a number of hydrogen bond donors and acceptors. You probably know this set of criteria as a Lipinski rule of 5. Also, the topological polar surface area of the molecule is added. So this is filter number 1 used very routinely in pharmaceutical chemistry to predict the oral bioavailability of the compound. So this is something what the students usually know from the very basic course of medicinal chemistry. There might be also other descriptors, for example, topological descriptors. Maybe Dr. Zitko explained in one of his lectures. So we use so-called topological indices or molecular fingerprints. So if we judge the molecule from the point of chemical structures, but we transfer the structure into numbers. For example, when a specific chemical feature is in the molecule involved, or we give the fingerprint 1. When there is not present in the structure, we assign the molecule 0. So these are topological descriptors, and we use also topographical descriptors. And you can see on the right side of the slide that there is a scaffold clustering of a set of compounds. And they are actually clustered on the type of scaffold, which means, for example, an aromatic feature or a bicycle in the molecule and grouped together. And the blue rectangles are the active compound. So we can explore what scaffold in the molecule is suitable to be present in the structure. So I'm checking the question now. What features allow a target to be druggable? So usually it might be the size of the protein and especially the binding site. It is suitable that the binding site is like a cavity, not on the surface, but inside the protein. Because when the binding site would be on the surface, so different molecules would be able to bind to it, and show some promiscuity, which is not intended. We desire to have the binding site in a cavity deeper in the protein, and only the suitable ligands are able to get there. Another question. Hit list is made of compounds that have similar structure and can bind. So the question is, hit list is made of compounds that have a similar structure and can bind to the protein target. It depends strongly on the input database. How huge and how diversified is the input database? So it's hard to say. It depends on the specific case. Hit list are all the active compounds from virtual screening. So we can use, for example, molecular docking and run through the protein a huge database of compounds. And the compounds that show some specific scoring and suitable pose in the binding site are the hits. But not all the hits can become drugs, because we can take into consideration also other parameters of the molecule, like solubility, Lipinski parameters, toxicity, selectivity, and so on. So I hope that I answered your question. So HIT is a compound that was active in a primary screening. It can be laboratory wet screening, wet assay, or it can be virtual screening in silico. For example, molecular docking or pharmacophore modeling and so on. So let me continue now. I would like to talk also about the IC50 value. So it's the half maximal inhibitory concentration of the compound, and we can judge the activity of specific compounds that inhibit specific targets. So the lower IC50, the more active compound. But in molecular modeling, we also the LE parameter, which is ligand efficiency. And it is defined as ratio of the affinity of a ligand divided by number of heavy atoms in the molecule. So it takes into consideration how big is the molecule of the ligand, of the binding compound also. So we take into consideration all heavy atoms in the molecule. These are all non-hydrogen atoms in the molecule in the terms of molecular modeling. So if you find somewhere in literature, in scientific articles, IC50 value, it might happen that you also find the ligand efficiency value. And here are again some examples for antiviral agents. So I have a short list or overview of drugs for whose the virtual methods were used during the development. So I have some structure-based drug design methods. So I will show some examples. Or for tirofiban, it was first drug for which the pharmacophore modeling was used. So now I would like to show the examples, not all of them, but some of them. So my first example is a structure-based drug design method involved in design of angiotensin converting enzyme inhibitors. So the very first compound was captopril, and the second one was enalapril, developed by different companies. The first one by Squibb, the second one by Merck. And the angiotensin converting enzyme is a membrane-bound enzyme, which is complicated to be crystallized. So the structure, the 3D structure of this protein was not known when the compounds were developed. It was only known that it is a zinc metalloproteinase, and it catalyzes the hydrolysis of dipeptide from the decapeptide angiotensin. So here you can see the reaction. Angiotensin 1 is the decapeptide, and the angiotensin converting enzyme splits dipeptide residue to get the angiotensin 2, which is a strong vasoconstrictor. When we inhibit angiotensin converting enzyme, we are able to decrease high blood pressure in patients with hypertension. So the 3D structure of angiotensin converting enzyme was not known. So the scientists started with a regular carboxypeptidase, which is also zinc metalloproteinase, but it only cleaves one amino acid residue, only one amino acid. It was known that the amino acid is, or this reaction is inhibited by benzyl succinic acid. So this was one of the starting points for development of inhibitors of angiotensin converting enzymes. And it was also known that there is an arginine residue that binds to the carboxy end of the peptide by ionic bond, and that zinc ion plays a role in the coordination of the carbonyl bond of the peptide bond. So you can see the scheme of the hydrolysis, and it was known that when benzyl succinic acid is bound to the carboxypeptidase binding site, it can inhibit the peptidic process. So the benzyl succinic acid was the first point, and there was another point, and it was the peptide teprotate that was known to be angiotensin converting enzyme inhibitor. It was found in a snake poison. It was isolated from Botrops jararaca, and peptide compound is a nonapeptide called teprotate. It was known that there is a proline on the C end of the peptide, or oligopeptide. So these two features, like the benzyl succinic acid and proline, were combined together, and the scientists started with the succinyl proline. However, you can see that the IC50 is several hundred micromolar, which is too high. So there was an optimization done by adding a small lipophilic substituent, and the activity was really increased. I mean, the IC50 was much lower, so the activity was higher. And by substitution of the carboxy group with a thiol, captopril as the first inhibitor of angiotensin converting enzyme was obtained. And there was another structural optimization that started with the succinylproline by the other company. A secondary amino group was mimicking the peptide bond. There was again a methyl added to fill in the S1 pocket in the protein, and the IC50 value was quite nice. However, the scientists performed further structural optimization, and they enlarged another substituent next to the amino group, bound to the carbon next to the amino group, and added phenylethyl substituent that was suitable for filling the S1 pocket that is much bigger than the S1 comma pocket. And you probably know that this is the structure of enalaprilate with nanomolar IC50, which is excellent result. However, the inhibitors of angiotensin converting enzymes are very poor bioavailable after oral administration. So they are all administered in form of ethyl esters. That usually have excellent oral bioavailability. So enalapril is actually prodrug because the ester moiety needs to be cleaved firstly to act later in the binding site of angiotensin converting enzyme, and the free carboxylic acid is important for the interaction with the zinc ion. I think that these compounds were developed in 80s. Later on, of course, the X-ray structure of complex with angiotensin converting enzyme was known. For example, for lisinopril, that has a amino group instead of methyl in enalapril. So this was the story of angiotensin converting enzymes. And let me continue with the design of thymidylate synthase inhibitors. There were two different approaches used. Firstly, the de novo drug design and the structure-based drug design, with the intention to develop anti-tumor, anti-cancer agents. So it is known that thymidylate synthase catalyzes the step from deoxyuridine monophosphate to deoxythymidine monophosphate by introduction of methyl substitution that is transported into the reaction by methylene tetrahydrofolate, which is the active form of tetrahydrofolate folic acid. So it brings actually the one carbon residue here in this position, transferring it to the deoxyuridine monophosphate to get the deoxythymidine monophosphate. So the human thymidylate synthetase structure was not known. So the scientists used a bacterial enzyme from E. coli that could be supplied in large amount and crystallize it with some known inhibitors. So the first one was fluorodeoxyuridylate, and the second one was the compound with CB code. And it was known, it was got to know that there is information on the binding mode. There is a ion dipole interaction, hydrogen bond interaction, and another hydrogen bond interaction via a water molecule. So the important amino acids residue were known that are involved in the interaction, and the scientists deleted the active compound and worked with the de novo method. So they suggested naphthalene moiety as the basic core, involved an lactame, which is internal or cyclic amidic moiety, and they further substituted the naphthalene with tertiary nitrogen. It's better to use nitrogen than carbon, because carbon would produce the chirality of the molecule. However, the nitrogen does not. So the nitrogen was desubstituted, and this part was introduced for water solubility increasement. Some interactions of this de novo compound were expected. However, the crystal structure did not confirm. There was bad interaction on the carbonyl of the lactame, so it was further optimized. There was actually modification on the lactame part of the structure, but also other substitution places, and docked into the thymidylate synthetase. And very active compound in the end was found. So instead of the lactam moiety, there was amidine moiety, and for water solubility, the morpholine was involved, which is a typical structural feature that enables to improve the water solubility. In addition, nitrogen is involved here in a sulfonamide moiety, which is also a hydrophilic part of the molecule. And this inhibitor was put into clinical trials. Another example, focuses on antiviral agent. So we are back in 80s of the last century, ritonavir and lopinavir that act as protease inhibitors, actually, it was known that in the active site there is C2 symmetry. So the company started to develop symmetric inhibitors that might have better selectivity, might be better recognized by viral proteases, and might have stronger binding. So the starting point was a reaction intermediate, which is the diol. So the left part of molecule was copied according to plane symmetry, and the dihydroxy moiety was reduced to only alcohol. So this was the starting point from the C2 symmetry. However, we have here a chiral center. And the molecule was optimized in combination of de novo design and structure-based drug design. So the target alcohol was not as much active, but when the valine moieties were added to the sites, the scientists obtained nanomolar IC50, and with further optimization, even units of nanomolar concentration in IC50. And they were able to get an X-ray of co-crystal of the inhibitor with the protease, and they started again to enlarge the middle part of the molecule with the hydroxy group, and started the symmetry on the carbon nitrogen bond. So they produced a target diol. And the optimization was quite long. The first compound was the target diol, but it was very badly water soluble. So what they did with the molecule, they exchanged the carbamate for urea moieties and exchanged the benzene rings for pyridine rings on both sides. This molecule was already very active. However, they tried to reduce one of the chiral centers to only one diol, which produced very active compound. And further optimization with putting back the carbamates produced compound that was not that did not have such a long or enough long half time, and they optimized further with thiazole and hydrophobic alkyl group that proved to be essential for the interaction with the target protease. This optimization with the thiazole moieties produced ritonavir with efficient concentration 30 nanomolar, and by putting some conformational restraint into the moiety, so that the urea moiety is bound within a cycle to restrain some movement and too many rotamers or conformers, lopinavir was produced with EC50 70 nanomolar. So this is an example of structural optimization, and the scientists always took into consideration water solubility of the compound, plasma half-time of the compound, metabolic stability of the compound, and also the oral bioavailability. I think that this middle compound was tested as intravenous antiviral agent. However, the final compounds were also orally bioavailable. So this was the example of de novo drug design and structure-based drug design of antiviral agent. We have an excellent story of kinase inhibitors of epidermal growth factor receptor that belongs to protein kinases. These are enzymes, membrane-bound enzymes with intracellular and extracellular moiety. And the function inside the cell is phosphorylation using the ATP. So most often are tyrosine kinases. However, we are able to inhibit also serine threonine kinases or histidine kinases. There are different types of inhibitors. The type 1 inhibits the active conformation by binding to the active site. And type 2 of inhibitors stabilizes the inactive conformation of the enzyme. So you have the particular examples. However, I think you do not need to memorize. What is important is the structure of the inhibitors, because on the right side, you can see in the figure ATP bind to the active site of kinase of epidermal growth factor. And you can see that there is a hydrogen bond acceptor interaction on the purine ring, hydrogen bond donor interaction on the amino group. There is a ribose pocket and 3 phosphate moieties stick out of the binding site in the cleft in the external part of the protein. So very first kinase inhibitor was gefitinib, developed by AstraZeneca in the year 2002. And the general structure is anilino-kinazolene structure, which mean phenylamino substitution on kinazolene moiety. It was known that the aminokinazolene moiety is essential for the binding, like mimicking the adenine in ATP. And it was known that on the phenylamino group are small lipophilic groups required. And there should be some electron donating substituents on the nitrogen bicycle. The first modification concerned the phenyl amino substituent, and it was necessary to block the hydroxylation of these metabolites. So fluoro and chloro substitution was introduced as metabolic blockade because chloro substituent is able with its size to mimic the methyl group, and fluoro substituent is in the size similar to hydrogen. So very active compound was obtained, and there was further optimization on the methoxy groups in position 6 and 7, where the electron donating groups were required, and there was morpholynyl propyl substituent, propyloxy substituent introduced morpholine again is added to increase the water solubility because it is stacked out of the active site and it contributes to water solubility of the molecule, and also it can decrease the energy penalty that would be required for desolvation of the molecule. So the part of the molecule with morpholine can stay solvated, and for binding is not necessary to desolvate. So we can see the molecule of gefitinib that was developed in this way, and another inhibitor of epidermal growth factor receptor, erlotinib. There is another inhibitor developed a few years later, introduced in the year 2007, and it's the lapatinib. It has the same target receptor. However, gefitinib and erlotinib are used for treatment of lung cancer, whereas lapatinib is used for treatment of breast cancer. You can also see the scheme, how the kinases function. So they are membrane bound with extracellular part and intracellular part. There are some ligands that are able to bind to the extracellular part, and after dimerization, which is sometimes required in the kinases, the activated dimerized receptor or kinase is able then to trigger some signaling cascades or pathways in the cell. So how the lapatinib was developed? It was again known that there is the hydrogen bonding region required. There is some restricted hydrophobic region, and there is some variable region. Below the main structure, you can see the particular compounds that were tested. You can see the variations either in the bicycle or in the side chain. And in the end, actually, there was a furyl introduced in the molecule in the side chain of quinazoline, and at the end, methan sulfonyl moiety was introduced in the lapatinib structure with excellent IC50 value below 1 micromolar. And you can see here the binding of lapatinib in the crystallographic complex. And you can see also some other compounds that were evaluated as candidates. And lapatinib is the first one. So you can see the variation instead of furyl, thiazole ring, and some other variation in the sulfo side chain. So lapatinib was the successful compound. I would like to talk now about pharmacophore-based screening. So pharmacophore modeling is also very often used method apart from molecular docking. It is more high throughput. I mean, in shorter time, we are able to screen huger compound databases if they are suitable or not. So this time, this is an example from a textbook where a histamine H3 receptor antagonist was developed. In the therapy, the H3 receptors are not yet influenced. However, they might be target in therapy of various CNS, central nervous system diseases. So in that time, no crystal structure for H3 receptor was known. It was only for H1 receptor that is used in allergy treatment. However, there were some ligands known, the structures of the ligands known, that antagonize the H3 receptors. On the right side, you can see the structures. So generally, there was a protonable nitrogen atom, either in a saturated ring or in aromatic ring. And in a specific distance, aromatic moiety. And linker with some hydrophilic functional groups. Based on a knowledge of 418 ligands with known affinities, the scientists tried to use homology modeling, but it did not help with the docking study. So they took only the compound 1, which is on the left side in the structure, and created pharmacophore model. So they employed the nitrogen in the molecule, aromatic part, also some other linker features, and van der Waals volume and excluded volume behind the molecule volume. And in the right side figure, you can see the final pharmacophore model, and they tested huge databases, Maybridge and World Drug Index database. Firstly, they used the filters like a Lipinski rule, and they ran the compounds through the pharmacophore model. And they restricted the number of the compounds to 2,600 and some other compounds. They then use the method leave one out filter. So they always left one of the pharmacophore feature and reduce the final number of the compounds to 320 that were used then for docking. And after docking, only 7 compounds were selected for testing. So they were prepared or boiled and then tested in a wet assay. Here one remark, what is the advantage of pharmacophore modeling? It can better limit the number of the compounds, and it can give higher novelty among the screen compounds, so lower similarity of the hits after screening. I would like to mention also homology modeling in the development of active compounds. So this is typically workflow. So please, when you study for the test, remember the workflow. Homology modeling is used when we do not know the 3D structure of a protein. However, we know the amino acid sequence, and based on a template, templates can be very relative or related protein where the 3D structure is known, we can use it as template and predict the 3D structure of the unknown protein. So we have to use or we have to find a similar protein in amino acid sequence and also in the structure. We have to explore the similarity, local similarity in the binding site, and check the similarity in function. The second step, when we find the template, we usually perform sequence alignment to find the position of the amino acids, to find the highest sequence identity. And then we are able to predict the 3D structure of the protein. And there are other two steps in the homology modeling, which is the model refinement, like checking if the structure is correct, and the last step is the model validation. So this is general workflow of homology modeling, and I have an example where it was used. It was used for constitutive androsten receptor structure prediction, which is a nuclear hormone receptor. Generally, probably you know that there are different nuclear receptors. For example, the steroid hormone receptors, vitamin D receptor, retinoic receptor, and so on. And these receptors have DNA binding domain, which is essential for binding to DNA in the cell nucleus. And there is a ligand binding domain, for example, for the steroid hormone, for vitamin D or vitamin A, and so on. So the first step is the binding of the ligand to the receptor, and then it is able to bind to DNA and somehow change development, differentiation, homeostasis, and reproduction of the cells. There are two types like homodimers, heterodimers among these receptors. And there are some other features in the binding like co-repressor and core activation. So as for the constitutive androsten receptor, the structure was not known, and the scientist knew only few ligands that were able to bind to this receptor. They started comparative or homology modeling, it's synonymous, And they took even two templates. The first one was nuclear vitamin D receptor, and the other one was nuclear pregnane X receptor. So they took these two templates and they determined the sequence identity to 40, from 40 to 50, which is sufficient. And they performed then sequence alignment. So in the first lines, you can see the one letter code amino acids for androsten, constitutive androsten receptor, pregnane receptor, and vitamin D receptor. And based on the comparison of these two known structures, they constructed using some software for homology modeling the structure of constitutive androsten receptor, which is here in the picture in gray. They checked also Ramachandran plot, you know what it is. It was discussed in the previous lectures, and they performed refinement. They also ran a molecular dynamics study, and they checked in docking program the binding of the two known ligands, which was clotrimazole and 3-paramethyl phenyl phosphate. So on the right side, you can see the structures of the ligands bound to the active site of the constitutive androsten receptor. And in the last step, they ran a virtual screening with the homology model, started with 85,000 molecules that were filtrated with the rule of file violation to 9,700 compounds. After docking, only 66 compounds were determined to be active, to bind well to the active site of the receptor, and they then tested the compounds in a wet assay, in a laboratory assay, and found 3 very active compounds that could be used in cholestasis treatment, theoretically, or neonatal jaundice. However, this receptor is called orphan receptor, so the purpose of influence of this receptor is not yet known. Later on, the X-ray structure of human and mouse constitutive androsten receptor was reported, and when it was superimposed with the homology model, the resolution 3.4 to 3.8 angstroms was found. However, when an uncertain region was excluded, the resolution was only 1.8 angstroms, which is excellent. So this was an example of homology modeling. And I would like to continue with a structure-based drug design of nitric oxide synthase. It is known that this enzyme catalyzes conversion of arginine to citrulline and nitric oxide, which is a vasodilating agent. And there are 3 isoforms of this enzyme. The first one is neuronal, the second one endothelial, and the third one is inducible inflammation nitric oxide synthase. So the compounds developed in this study could be anti-inflammatory agents. acting via inhibition of inducible nitric oxide synthase. It was known that the inhibitors contain basic amidine, mimicking the arginine moiety and interacting with the glutamic acid in the binding site. So the Swedish company AstraZeneca started a campaign on development of these inhibitors and determine some co-crystal structures of kinazolines, aminopyridines, and thienooxazepines. So the 3D structures of the complexes help to decipher the suitable structure for binding. It was found that glutamic acid and glutamine are essential for the binding, and there is also hem at the bottom of the cavity that can support the binding. So they found, the scientists found many inhibitors. They also tested the selectivity against the 3 isoforms of the enzyme, and compound 8 was the best one, maybe 9 also. Sorry, and compound 11, you can see there here is a PDB code, and it is super selective against the inducible nitric oxide synthase. And you can also see that the amino group in the structure was really essential in the binding to the glutamic acid. So it was development of AstraZeneca, and we have a real drug where the fragment-based drug design was used, which is the vemurafenib. Indicated for treatment of metastatic melanoma, and it is the first example of fragment-based drug design used by Plexicon company and Genentech company. So the company started with 20,000 scaffolds, and you know that the fragments are usually very small molecules, maybe sometimes up to 250 daltons. In this case, it was up to 300 daltons, and screened against kinases. And it afforded the compound 29, which is an azaindole that bound well to the binding site, and via adding some substitutions in the molecule, the final structure grew into the compound 32, which is the clinically used vemurafenib for treatment of melanoma cancer. So the company did not use real ligands, but only very small fragments, and used then the method of growing the molecule into real large of clinically used drug. I would like to talk now about Aleglitazar, which was developed by Boehringer Ingelheim, Mannheim, a German company, and they claimed a campaign in the year 2000 for peroxisome proliferator activated receptors in the mean of agonists of these receptors. It is known that there are three isoforms. It's PPAR alpha, PPAR delta, called also beta, and PPAR gamma. And the company wanted to get co-agonist that would be able to influence PPAR alpha that are present in different tissues like muscles, kidney, arterial cells with a high rate of mitochondrial fatty acid oxidation, and influencing and agonizing also the PPAR gamma that are present in adipose tissue and are able to activate the fatty acid usage. So Boehringer started with several points because there were plenty of information known from the competitors, like the other pharma companies, for example, from GlaxoSmithKline on rosiglitazone, then from Takeda, Eli Lilly on pioglitazone, and from AstraZeneca on tesaglitazar, which was later stopped due to some adverse effect. However, the German company knew the information on the binding mode in the active site of PPAR receptors, and all the amino acids involved in the interaction were known. So it means in the structure, there was essential carboxy group that was able to bind to create the hydrogen bond to histidine and to other tyrosines. Farglitazar was another structure found as the strongest gamma binder. So they took a lesson also from the structure of farglitazar, and the benzothiophene linker was inspired by edaglitazar. So we can see the structural feature here, and also the activities against PPAR alpha and gamma. And Boehringer Ingelheim company was able to develop, well, I would guess this is a record, excellent time, only of 2 weeks, to synthesize aleglitazar that became then efficient drug. So here you can see the structure, you can see also the binding mode in human PPAR gamma receptor and PPAR alpha. It has the typical banana shape carboxy group for binding benzothiophene moiety, and the molecule is somehow partly broken in the middle in the region of the flexible linker. So this was a very nice example of structure-based drug design. And let's continue with antiviral agents. This was again example of structure-based drug design, how the oseltamivir and zanamivir were developed. The first one by GlaxoSmithKline company in 90s, and oseltamivir by Gilead Sciences. So the structure of the target enzyme, which is neuroaminidase, was known, and also the binding of sialic acid analogs like intermediates in the physiological pathways was known. So due to the binding mode of acetyl neuraminic acid, which is the acetyl sialic acid, the scientists retained in the structure the essential features, and they obtained zanamivir and oseltamivir only by modification or removal of the hydroxy group and modification of the side chain in oseltamivir. So this time for development of zanamivir and oseltamivir, the knowledge of neuroaminidase 3D structure was helpful. And I have last 4 examples from Roche company. The first one focused on dipeptidyl peptidase 4 inhibitors that could be useful in the treatment of diabetes mellitus type 2. So again, the company claimed a campaign on finding active compounds, and the basic core of these compounds was aminobenzoquinolysine moiety, and they tested different side chains with the best acting compound. I think it was number 9 with the 7 nanomolar IC50. I will go quickly through these, through the other examples. This is again a potential treatment of diabetes and atherosclerosis targets by influencing fatty acid binding protein 4 and 5. So relatively simple structure with quinoline basic core, varying the substituents in position 2, getting differently selective derivatives. Third example from Roche computational laboratory is the inhibition of cathepsin S and cathepsin L, which would be useful in the treatment of glomerular diseases of the kidney. So the structures are this time a little bit more complicated, but you can see the IC50 values and the binding modes in the active site of the particular cathepsins. And last but not least was the searching for inhibitors of beta tryptase, that which is a serine protease expressed in mast cells and they can play a role in treatment of airway inflammation or allergic responses. So again, some active compounds because the 3D structure is known. So I think I gave you a lot of examples, and I would like also to summarize some other important properties of drug candidates. We have seen some IC50 values, binding modes in the active sites. We have seen the selectivity. However, the scientists have to include also other items. For example, water solubility, which is essential for transportation in the body, but also for the drug delivery, for example, for solution administration or drops administration. The lipophilicity, expressed as log P or log D, is essential for crossing the membranes. It is included in Lipinski rule parameters. Then microsomal stability can predict how quickly the compound will be cleared, so we can predict the half-time of the compound in the body. Then inhibition of cytochrome, P450, we can predict the metabolization of the drug and also some potential toxicity. In CACO2 cells, we can estimate the permeability across intestine. It is essential for compounds with a basic character that are usually absorbed from intestine. We should take into consideration also the interaction with a glycoprotein P, which functions as efflux pump in some resistant, for example, cancer cells. Or we can judge the hepatotoxicity in relation to HepG2 cell line inhibition. And generally, we can predict the cytotoxicity in any other cell line, for example, some healthy human cell lines. So in the right figure, you can see the cascade, how the parameters are take into consideration during the development of the lead compound, then into a candidate compound. And in the design, we have always to synthesize the compounds, test them in real time in the laboratory, analyze, and then we can use again the in silico design. We are able to modify the affinity and selectivity by filling of lipophilic pockets in the binding site of the proteins, adding polar substituents that are essential for binding, for the target interaction, and also for water solubility. We use the scaffold hopping method, where we can change different aromatic features for some analogs. We can also transfer structure activity relationship. We can perform conformation analysis, put some conformational restraints into the molecule, and perform also molecular overlays. And there are some other methods we mentioned with vemurafenib, the fragment-based drug design. There is also space to design compounds inhibiting protein-protein interactions, which is typical for the compounds from the group of molecular glues. This is a very interesting group of compounds. We can take into consideration also allosteric modulators. And in case we have enhanced some active compounds and we do not know the target that is influenced by the compounds. It is, for example, the case of our research group. In some compounds, we know that there are great antimycobacterial agents. They are able to inhibit the growth of mycobacteria. So would be potential anti-tuberculosis drugs. However, sometimes we have to fish the target, find the suitable target based on similarity search, machine learning, or some docking processes. And that would be all from my side today. I hope you survived the lecture. And I can see some questions in the chat from Salaha. So okay, I will start from the beginning. There is a question on thymidylate synthase inhibitor development. They started with de novo methods, old non-molecular modeling technique, and they then did lead optimization with structure new method. Yeah, the scientists combine different type of methods. They combine, for example, pharmacophore modeling with molecular docking, or molecular docking with molecular dynamic. They also combine the de novo design method. However, the design of the compounds from this approach needs to be then verified in the structure-based drug design method. Another example, how do they test each new molecule after each structural optimization? Do they test them on animal models? Usually they start with the target assay. So they need an laboratory assay where they test or screen the inhibition or agonization of the target protein, and only the very prospective compounds come then into animal models, because it's very expensive to test all the compounds in animal models. However, there are some computational engines also how to predict metabolization of the drug, how to predict the toxicological profile of the drug, but we did not involve these methods into the discourse. So definitely before registration, before getting to the market, the drugs need to be tested in animal models, and especially different type of animal. Another question, is there a structural reason for the fact that lapatinib is for breast cancer? Actually, there are different types of epidermal growth factor receptors. So it depends on the type of the cancer, what type of this receptor it produces. So we can target the specific receptor with specific compound. However, among the kinase inhibitors, there are compounds that are more selective against specific kinases, and there are compounds that are generally used. They have the multiple effects, or they are able to inhibit different type of kinases. Okay, last question. For the constitutive androsten receptor protein modeling, did they merge the pregnant X receptor and vitamin D proteins together? So they spliced together the amino acid structure that were most similar. They performed, so they used two templates, and they performed alignment with the two proteins, with the pregnant X receptor and with the vitamin D receptor. So they used not only one template, but two templates, and performed the alignment of the amino acids in an optimal way to have the highest sequence identity. And they then constructed the 3D models based on the knowledge of 3D structure of these two template proteins. And then, of course, they refined the model and they validated the model.